Okay, so it's Thursday. What we're going to do today is we're going to kind of finish up these PowerPoint notes and go through a couple other things. Tomorrow you guys will be working on something called cladograms, which kind of goes along with classifications, another way to go through and classify things based on traits, and what we're looking at is more or less the evidence of evolution and how closely related organisms are based on structures and amino acids, sequences and other things like that that they can have in common with one another to see how closely related they actually are. But today what we have, we're going to finish up some stuff on genetics with evolution. Tomorrow, like I said, will be cladograms. We'll kind of go through and review. You'll probably get a little bit of a review sheet tomorrow. Maybe I'll send it out over the weekend. Nothing that you really have to do, but then you can kind of work on it and ask me questions on Tuesday because Monday is that flex day. And we'll probably have a test, your last test, on evolution on Wednesday, since that is now our day 2, and I meet all my classes on that particular day for you guys. So, if you guys think back to genetics a little bit, we did a probability activity where you guys flipped coins, and that's kind of what this first little slide looks like. So besides evolution being caused by natural selection, evolution can also be caused by chance. So flip a coin. What is the chance that it lands on heads or it lands on tails? And you guys did that. You did that 10 times, and you kind of calculated out what the probability of heads versus tails would be. You did it 100 times. What's the probability of heads versus tails? And you should see, the more times you did it, the more flips that you had, you probably got closer to that 50-50 mark of heads versus tails. So think about it this way. If you flip a coin 1,000 times, let's say 700 times it's going to land on heads, 300 times it lands on tails. You would be suspicious with that because you should expect a 50-50 shot between those two. If you flip a coin 10 times, 7 times lands on heads, 3 times lands on tails, you'd be like, yeah, okay, I can kind of understand that. And the reason is it is a smaller sample size. So the smaller the sample size, the more likely the chance alone will cause deviations from expected results. So if I'm looking at that smaller sample size, I only did it 10 times, yeah, I could see that 7 times on heads and 3 times on tails. But if I did it 1,000 times and I had 700 and 300, I would go, oh, maybe I wasn't flipping the right way. Maybe I was just kind of throwing up in the air. Maybe I didn't mark them down correctly. So know that the smaller the population, the smaller the sample size, the more likely we're going to see chance play a role in what we see with evolution. So a couple things can happen here. And the first term that you guys have here is what is known as genetic drift. So still thinking about genetics. If certain genes, by chance, show up more often in a small population, that trait becomes more common in that small group. So we see this a lot more with endangered species, and the reason is because their populations are a lot smaller. So they're passing the same genes back and forth. So if we have this small group, we're going to see these traits that we may not see in larger populations get passed along. So we say that it is more common in small groups than in species as a whole. We see this with certain genetic disorders. So things like, if anybody did for their genetic disorder brochure, maple syrup urine disease. It's one of the ones that affects Amish and Mennonites. Think about it. They are a smaller population of people. They usually marry within their same group. They have the children within the same group. They live within the same group. And they're not having influences of other genes. So they are a much smaller, what we consider population within them. So they're just kind of passing those same genes back and forth. They don't have a very large gene pool or variation within those genes. This microevolution, micro, small, we're talking about small groups, we see it a lot with endangered species, can be caused by 2 things. The first one is called founder effect, and you guys are going to see a little video and Edpuzzle that goes along with this idea of chance and genetic drift and founder effect. So keep that in mind as you go through. So founder effect. A small group colonizes a new area. So let's say a population of people decide that they're going to go off on a boat and they're going to live on that island that's 100 miles offshore and they're going to stay there. Well, whatever genes are within that 100 people may not be a fair representation of the genes from the group or the colony that they originated from. So if this small group colonizes a new area, we now have a small gene pool. And this can occur with plants, it can occur with animals. Little seeds come off the plant, they kind of blow off in the wind, maybe they land someplace else, and we have that founder effect. We may change that population, that new population is going to start because the genes that we have there may be different than what we see with that larger colony that they originated from. And this is what we see with genetic disorders. And we have a loss of variation here because we have such a small gene pool. Remember, small gene pools are not good. We want a variety or a variation within those genes. Next one is called bottleneck effect. And this is more like an event occurs. So maybe an earthquake comes along, a tsunami comes along, a forest fire comes along, and drastically reduces the population size. So something comes along and wipes out a lot of the organisms within that particular area, within that particular population. Those are left by chance. It's not like they were smarter or they blended in better. The chance that they survive was just that. It's chance, and evolution is just that. So as they go through, we have a loss of genetic diversity, then the whole species. This is not good. Only a small group survives. The population shrinks. Genes get lost. They might not be good genes that those organisms are left with. Think about it this way. You're walking down the sidewalk and you see this big thing of ants or bugs on the sidewalk, a big colony of them, and you decide to step on them or you roll your bike over them and you crush about 3/4 of them. That's a bottleneck effect. So those quarter that are left, those quarter of organisms that are left, they have a smaller gene pool. You lost a lot of the genetic variation. So that genetic variation may have died out with those 3/4. You may only have a few genes within that variation left with the quarter of them that are left. So we see this microevolution and this idea that evolution can also be caused by chance with this. Not only natural selection, but a lot of times we see it with only your smaller populations. Moving on. So that kind of wraps up some stuff there. We want to go back and talk a little bit about early Earth. And we've talked about this before when we did bacteria. We talked about how bacteria theory-wise was the first form of life on Earth. We said the Earth formed about 4.6 billion years ago. So it first formed, and the conditions on the planet at that particular point were really harsh. There was no oxygen. There were some deadly gases. It was real torrential conditions. Our first form of life came about 1 billion years later. So 3.5 billion years ago. And we actually have microfossil evidence of particular bacteria from 3.5 billion years ago. So as organisms evolve and change, about 2.1 billion years ago, we go from those prokaryotic cells to those eukaryotic cells. They have a nucleus, they have mitochondria, they have chloroplast, and it brings us to the theory, that endosymbiotic theory by Lynn Margulis, who said, okay, well, how did we go from this stage, prokaryotic cells, to this stage, eukaryotic cells? Where did we get those organelles from? And that's what the endosymbiotic theory states. And we've done this a few times. We did it with photosynthesis, we did it with cell respiration, we talked about it with bacteria. This is basically how mitochondria and chloroplast, those organelles in eukaryotic cells, evolved. And we said they evolved from prokaryotic cells. So remember, bacteria engulfed or gobbled up endosymbiosis, active transport. They gobbled them up, they brought them in, and they survived. And every time they replicated, the organelles, or these little bacteria that eventually formed organelles, also replicated. And over time, they started to utilize the sunlight in order to make food or break down food to get energy. And they evolved, and it wasn't like the next day, they became mitochondria and chloroplast. We're talking thousands and thousands and thousands of years for them to evolve into these particular structures. So if we think about those conditions on early Earth, and there's still a lot of, ideas out there, and they're still saying, how could this happen if the conditions were like this? Where did life come from on this particular planet? Two scientists, Miller and Urey experiment, goes through, and they basically prove that organic molecules formed spontaneously under conditions that were seen on early Earth. So they were able to set up in a little experiment. They said, okay, well, on early Earth, and we have, ideas of what was there gas-wise because it's kind of trapped in certain things, ammonia, hydrogen gas, methane. What there wasn't was any oxygen. There were some inorganic compounds. We call this kind of this primordial soup. So they threw all this stuff, this water, and made some water vapor, and we threw everything in there, and they gave it a spark. And this spark was supposed to be lightning. And it's this lightning that kind of gave that energy. And it changed what they had in there, those gases, and made this primordial soup. It made this organic compounds. We had amino acids. They had some RNA in there. And there was some iron in there. And they're saying from that, it started to evolve to form those prokaryotes, those archaeobacteria, first form of life on Earth. We go to endosymbiotic theory, where we start to produce oxygen. Because remember, once we get chloroplasts, those chloroplasts are going through photosynthesis, they're releasing oxygen as a byproduct. and they're putting oxygen in the atmosphere, which is going to allow other eukaryotic organisms to survive. Finishing up our slides that we have here. The last little one says scientists believe that land masses were once joined. So Pangea, we had that huge landmass, and over time it started to break apart and things started moving. And that's why we can say organisms are kind of similar on different continents, but they're not going to be the same because evolution occurred, divergent evolution. So that giant landmass that we may have had to start out with had one type of organism. As the land masses began to separate, they had to evolve to the area in which they lived on, the food sources that were there, the temperature that was there, or the climate that was there, their predators, how they build homes, how do they get their mates. Organisms diverged from common ancestors. So besides genetics, we can also say that natural selection can alter populations in 3 ways. And this is kind of how we see how natural selection changes. So there are 3 that we have here. We have stabilizing, we have directional, and then you guys will also see disruptive. So if we look at this, stabilizing, what we have is the average phenotype. So if we think about a population, and within that population there's variations. The average phenotype is selected for, and the extreme phenotypes are selected against. So you have some of my extreme phenotypes on both sides here. The middle phenotype is the good one, and that's the one that's going to survive better, and we're going to see more of them. So let's think about it this way. Stabilizing. Robins typically lay 4 eggs within their clutch. They lay 4 eggs that are going to hatch into babies. Larger clutches may result in malnourished chicks. So if that robin lays 6, 7, 8, those chicks aren't going to be as healthy. That would be an extreme phenotype. Those chicks won't be as healthy. Mom can't feed all of them. So they're not going to have a very good chance of surviving. Smaller clutch size may result in no viable offspring. Maybe my mom only lays 1 or 2 eggs. Another one of my extreme phenotypes. Maybe those eggs don't hatch, or maybe those get eaten by other organisms. If mom lays 4 eggs, that's the average phenotype, the average number of offspring that robin should lay. She can raise them right. She gives enough food to them. If one of them gets picked off by a predator, she still has 3 more. So that's my stabilizing one, where my average phenotype is the one that's selected for. That is the good. Directional, on the other hand, is where it can change. So the extreme phenotype is favorable and the population shifts. And the best thing to think about this is with peppered moths. So we have light colored peppered moths, we have dark colored peppered moths. So the light colored pepper moths is one extreme, the dark colored pepper moths is the other extreme. So during the Industrial Revolution, there were a lot of light colored peppered moths, and they blended in with the trees really well. The dark ones did not blend in, so they got eaten off a lot. So if you were a light colored pepper moth and you were sticking to a tree, the lichens on the tree or the bark on the tree was also very light in color. So camouflage, they blended in. The population increases. The dark ones get eaten off. Well, the Industrial Revolution comes along and we start to do a lot of industry, and we're using a lot of factories, and we're putting a lot of soot in the air. And it starts to get picked up on the trees. So the trees begin to change, and the bark on the trees goes from a light color to a dark color. So now the low number of dark colored moths, they now blend in better. And as they blend in better, they're camouflaged. And they're going to start reproducing more and producing more of that particular variation. My light colored ones, however, they don't blend in anymore. They stick out. So what happens is they're going to go through and they get eaten off. So it's going to shift back and forth depending upon what's going on in the particular environment. So with pepper moths, it started out light. Those were the favorable ones. As the Industrial Revolution goes on, the dark ones become favorable and the light ones are going to end up dying out. This can constantly go on and shift back and forth to these extreme phenotypes. Disruptive selection. This one's a little bit more rare. So the average phenotype is selected against, and the extreme phenotypes are favorable. But what can happen is that it can produce 2 separate populations eventually. So in a hypothetical population, we have gray and Himalayan, which is gray and white rabbits, are able to better blend into rocky environments than the white rabbits. So these 2 actually blend in better. So within a population, there's variation. So mom has a couple bunnies, one's white and gray, one's gray. Well, this one blends in pretty well. This one blends in pretty well. Give me 1 second. Okay, sorry about that. That's what happens when the dogs go walking by and freak my dogs out. Okay, so disruptive, like we were talking about. Population, we have variation. Mom has some bunnies. We have white ones. We have white and gray ones. We have gray ones. Well, these guys blend in pretty well. These guys blend in pretty well to their rocky environment. These guys kind of stick out like a sore thumb. So in comes the hawks or the eagles, picking out those white ones that are not camouflaged in. These two variations survive. The genes that are within these, eventually over thousands of years, we can actually go through and produce new populations. So my medium phenotype is the one that's basically going to end up dying out, and my extreme phenotypes are the ones that are going to create those two separate populations. The last little scientist that I want to talk about is called Hardy Weinberg. So Hardy and Weinberg, and at this time of the 1800s, 1900s, everybody's kind of jumping on the bandwagon. And they all want to go in through and talk about this idea of evolution and natural selection and where did these organisms come from and how did we evolve? So Hardy Weinberg comes up with kind of this mathematical formula, and I'm not going to go through and ask you guys to calculate anything in terms of alleles and gene frequencies and things like that. But you should realize that Hardy Weinberg talk about evolution is minor changes within a species from generation to generation, which eventually can result in new species. And this is kind of what Darwin said with natural selection. So we talk about allele frequencies, and allele frequencies in a population will remain constant unless one or more factors causes those frequencies to change, and that's called genetic equilibrium. So there are 5 conditions for genetic equilibrium, and if you think about it, they're never ever met. They're saying, oh, if we meet these, the allele frequency is never going to change. We're not going to see new organisms form. Well, we can't stop these things from happening. So just by any of these things happening, we know that evolution is going to occur. So if we think about a population, Hardy Weinberg said, oh, we can't have random mating. Organisms will only mate with other organisms within their little group, and they will only mate with those and not mate or spread their genes with other populations. They said that you have to have very large populations, and the reason is because alleles are lost in small populations. No movement in or out of populations. Well, okay, I have this rabbit. I have a whole population of rabbits. Maybe some of them go off. They join another little rabbit family in the woods, and they now become part of that. Organisms don't stay within their population. Think about humans even. We don't stay within the same country. We go to other countries. We marry people from other countries, other states, things like that. So we have movement in and out of populations. Hardy Weinberg said, okay, for this to occur, meaning our allele frequencies are going to change, we need no mutations. Well, we know we can't stop mutations. Mutations are always going to happen. And when we have mutations, we change the allele frequency. And eventually it's going to change populations, leading to evolution. And he also said natural selection does not occur. Nature does not select traits that are favorable. Well, we absolutely know it is. So, they said that evolution will not occur if in a population if the 5 conditions are met. We know these conditions are met. We know that evolution is occurring. So if a population is in Hardy-Weinberg equilibrium, the genetic makeup of a population remains constant unless the factors are operating, meaning evolution. And we know that. We don't see it day to day. We don't see it from generation to generation. But if we look at many generations along the way, we can see that these conditions are not met. We can see that evolution is occurring. We can see that this whole idea of allele frequencies and genetic equilibrium is not remaining constant. So evolution is a change in frequencies of alleles in the gene pool of a population. And like I said, there is a whole formula-based thing that we can look at here. And I'm not going to have you guys, we can actually do it with alleles, who's dominant, who's recessive. And we can kind of calculate in a population how many people are homozygous dominant, how many people are heterozygous, how many people are homozygous recessive. And by comparing genotype frequencies for each generation, one can see if evolution has occurred, and not only that, but the rate in which it will occur. So we could go through and calculate this out. If you guys take AP Bio, you guys will see a lot more of this and do a little bit more with the Hardy-Weinberg. That's why I didn't want to just be a surprise to you guys. You guys should know what it is. But the 2 scientists that talked about allele frequencies and the conditions. And we know that these 5 conditions are basically never met, which means that evolution is constantly occurring. Okay, so you guys will see a couple little videos that go along with this stuff, a couple little Ed puzzles. Tomorrow we'll go through cladograms, finish that up, and then I'll be sending out kind of a little bit of a review sheet. So hang on to these videos, hang on to some notes. If you guys maybe like wrote some stuff down, make sure you have a copy of those PowerPoint notes there, so that when you guys have the test, you guys are able to go through and kind of utilize them. Let me know what questions you have. Monday, I will absolutely hold office hours for you guys, because that's our flex day is Monday. And we can go through and see what questions you guys may have on that. All right, so have a great Thursday. We'll talk more later. Bye.